Welcome back to the deep dive. Today, we are not just talking about medicine, we are talking about, well, a siege.
That's a great way to put it.
We are looking at a topic that represents perhaps the single greatest intellectual triumph of modern pharmacology, but also its most persistent, high-stakes stalemate. We're talking about antiretroviral therapy, or RT.
It really is the ultimate chess match. If you look at the history of medicine, we usually find a bug, we find a poison that kills the bug, and we win.
Right. Simple as that.
Penicillin kills bacteria,
Yeah.
quinine kills malaria. But with HIV, the rules just completely changed. We found a bug that we couldn't just poison because it weaves itself into the very fabric of our own genetics.
That's the central tension we're unpacking today, isn't it? We're dealing with a virus that is diabolical in a very specific, almost engineered way. It integrates into our DNA and it mutates faster than almost anything else on the planet.
Which presents this incredible paradox. We cannot currently cure it. We can't scrub it from the body completely, but we can control it so precisely with such, molecular engineering that a patient diagnosed today can live an essentially normal lifespan.
So to help us navigate this siege, we've pulled the heavy iron from the bookshelf. We are deep in the stack today. We have the section on antiviral agents from Ketsum, which gives us that essential clinical scaffolding, the drug classes, the protocols, the things you need on the wards.
The how-to guide.
Exactly. And backing that up, we have the Bible itself, Goodman and Gilman's, specifically the chapter on antiretroviral agents and treatment of HIV infection for the heavy molecular lifting and the systems level math.
And you really need both. You do. Ketsum tells you what to prescribe, but Goodman and Gilman explains why it works, or, terrifyingly, why it sometimes fails. It gives us the math behind the resistance, which is really the engine of this whole story.
So let's set the mission for this deep dive. We don't want to just rattle off a list of drug names. That's boring. And frankly, you can look that up on an app. We want to understand HIV pharmacology as an intervention in a living, evolving system.
Right.
We want to understand how we turn a fatal, unchecked replication cycle into a managed, chronic quietude.
Exactly. We need to look at this through the lens of durable containment. We're going to map the drugs to the viral life cycle, sure, but we need to understand the strategy, the logic of combinations, the concept of genetic barriers, and why adherence is actually a pharmacological variable, not just, a behavioral one.
Before we jump into the weeds, here are the key takeaways we want you to have mastered by the end of this session. First, life cycle targeting. Knowing exactly where we hit the virus entry, reverse transcription, integration, and processing.
Second is combination logic. You need to understand why multi-class regimens are the only way to prevent resistance. It's all about the math.
Third, genetic barriers. This is a big one. Why are some drugs forgiving of a missed dose while others break instantly?
And fourth, the adherence reality. We'll explain why missed doses are the true enemy of control, and why taking your pills 50% of the time is actually worse than taking them 0% of the time.
And finally, the public health lens. Viral suppression equals prevention. The concept of UU, undetectable, equals untransmittable.
Exactly. So let's get into the problem itself.
Section 1, the problem. Why is HIV so exceptionally difficult to beat? Because on the surface, a virus is just a packet of genetic material. Why is this one so special?
To understand that, you have to look at the retro in retrovirus. This is fundamental biology. The central dogma of life for you, for me, for a bacteria, is that information flows one way.
Okay.
DNA makes RNA, and RNA makes protein. DNA is the master copy kept safe in the vault, the nucleus.
Right. It's the sacred blueprint.
HIV flips the script completely. It carries its code as RNA, but when it breaches your cell, it brings along a specialized enzyme called reverse transcriptase, and it does exactly what it sounds like. It takes that RNA and writes it backwards into DNA.
Hence, retrovirus, going backwards.
Precisely. But here is the kicker. And this is the number from Goodman and Gilman that should stop everyone in their tracks. This reverse transcriptase enzyme. It's sloppy. It is a terrible typist.
What do you mean by sloppy?
It has no proofreading mechanism. When your cells copy your DNA, your enzymes are constantly checking their work, fixing typos. Reverse transcriptase just doesn't. It makes about one mistake for every 10,000 base pairs it copies.
That sounds like a lot of mistakes.
It's a huge error rate. Now, combine that with the replication rate. In an untreated patient, the body is churning out roughly 10 billion with a B-infectious viral particles every single day.
10 billion copies a day with a high typo rate.
Do the math. If you are making billions of copies and making random mistakes in every single one, statistically, by the time a patient walks into the clinic for their diagnosis, Every single possible point mutation in the viral genome likely already exists somewhere in their body.
That's a terrifying thought. You're saying the virus has essentially pre-calculated the answer to the drugs before we've even administered them?
In a probabilistic sense, yes. The library of mutations is already open and on the shelf. This is what we call the quasi-species. An infected person doesn't have one single virus. They have a swarm of billions of slightly different viruses, a cloud of variants.
A cloud of possibilities. And that brings us to the integration trap.
Yes?
So once it makes that sloppy DNA, copy typos and all, what does it do with it?
It marches it right into the nucleus of your T cell. It uses another enzyme called integrase to literally snip your human DNA open and paste the viral code right into the middle of your chromosome.
So it's not just an invader in the room. It has written its name into the building's blueprints.
Precisely. Goodman and Gilman calls this the proviral archive. And this is the fundamental reason we can't cure it yet. Even if our drugs stop every single new virus from being made, that original blueprint is still sitting there, archived in your long-lived memory T cells, waiting, silent, indefinitely.
So the drugs we have are virus static. They stop the churning factory. But they aren't curative because they can't go into the archive and excise that blueprint.
Correct. We are holding the line. We are preventing the archive from opening and making new copies. But if you stop the drugs, even for a few days, the machinery wakes up, the archive opens, and the virus comes roaring back.
And the cost of that roar is the immune system itself. Ketzel makes a big point of the CD4 count.
Right. This is the clinical consequence of all that molecular machinery. The virus specifically targets and kills CD4 positive T lymphocytes. These are the generals of the immune system. They coordinate the entire defense.
So as the viral load spikes,
the CD4 count crashes. And when you drop below a certain threshold, around 200 cells per cubic millimeter, you lose the ability to fight off everyday pathogens. That's the definition of AIDS. You don't die of HIV, you die of a fungus like pneumocystis, or a bacteria that a healthy body wouldn't even notice.
So the goal is simple. Suppress replication completely. If the virus can't copy itself, it can't kill CD4 cells, and the immune system can slowly recover.
That's the whole game. Get the viral load to undetectable.
But this brings us back to that terrifying math you mentioned. If the mutations are already there in that quasi-species cloud, why doesn't the virus just immediately become resistant to whatever pill we give?
Now we're getting to strategy. This is the logic of combination therapy. This is the 3-drug rule.
Section 2, the math of combination therapy. Walk us through this. If we have these incredibly potent drugs, why can't we just take the single best one we have?
Because it's a probability game and a numbers game. Let's say you have a great drug, we'll call it drug A.
Yeah.
And let's say the chance of a single virus having this specific mutation to beat drug A is 1 in a million.
Okay, that seems pretty rare.
It is. But you don't have one virus. You have 10 billion viruses. So 1 in a million odds with 10 billion viruses means you already have, on day 1, about 10,000 viral particles in your body that are naturally immune to drug A.
Right. They just happen to have the right typo.
They won the genetic lottery.
Yeah.
So if you give monotherapy, you kill the other 99.9999%. But those 10,000 survivors, they're now free to replicate without any competition. Within a couple of weeks, your patient is full of a virus that is completely resistant to drug A.
Okay, so monotherapy is just a resistance factory. You're actively selecting for the toughest bug in the swarm.
You're running an evolutionary boot camp.
Right.
But now let's say you use drug A, drug B, and drug C simultaneously. And crucially, they all have to attack different parts of the virus.
Different mechanisms, different locks on the door.
Exactly. For a virus to survive, it would need to have the mutation for drug A, plus the mutation for drug B, plus the mutation for drug C, all in the same single particle, all at the same time.
So you're not adding the probabilities, you're multiplying them.
You're multiplying them. So it becomes 1 in a million times 1 in a million times 1 in a million.
That's 1 in a quintillion.
Yes. The number is 10 to the negative 18th. That is so astronomically rare that even with billions of viruses, that supervirus just doesn't exist. Not on this planet. That is how we win. We overwhelm the math.
That's incredible. It's like trying to guess the combination to 3 different safes at the same time. The virus just can't do it fast enough.
Provided, and this is the huge caveat we'll keep coming back to, provided the patient takes the meds every single day and keeps the pressure on all 3 locks simultaneously.
The adherence cliff. We'll get there. So, we understand the strategy. Hit it with 3 different weapons at once. Now we need to open the toolkit and understand the weapons themselves.
Let's do it.
Section 3, the toolkit. And we have to start with the NRTIs. the nucleoside reverse transcriptase inhibitors, the historical backbone of essentially every regimen.
These are the classics, the OGs of HIV therapy. You'll recognize names like zidovudine or AZT, tenofovir and tricidobine, abacavir. The mechanism here is honestly beautiful in its simplicity. It's a Trojan horse strategy.
Help us visualize this. How does a Trojan horse work at a molecular level?
Well, the virus is in the cell, trying to build that DNA chain we talked about. It's using its reverse transcriptase enzyme to do the building. It's grabbing floating building blocks, nucleosides, the A's, T's, C's, and G's, and stringing them together like beads on a necklace.
Okay, a construction site.
NRTIs are fake building blocks. They are chemically engineered to look almost exactly like the natural food the virus wants. So the enzyme sees it, thinks, oh, here's a thymidine, and grabs it.
So the virus grabs the drug thinking it's a normal piece of DNA?
You're right. It snaps it into the growing chain. But here's the trap. These drugs are cleverly modified. They are missing a critical connection point, specifically the 3' hydroxyl group.
That's the little hook for the next bead on the necklace.
Exactly. It's a brick with no mortar on top. It's a Lego with the top stud filed off. Once the virus places that drug into the chain, it reaches for the next brick to add on.
Yeah.
And it can't. There's nowhere to attach it. The chain terminates. Construction halts.
Dead in the water. That's why they're called chain terminators.
But there is a nuance here that both Katzun and Goodman and Gilman emphasize, and it's vital for understanding the pharmacology. These drugs don't work right out of the bottle. They are prodrugs.
Meaning the pill itself is inactive.
Yes. When you swallow an abacavir or a lamivudine, it floats into your cells. But it can't do anything yet. It's inert. It has to be phosphorylated 3 times. It needs 3 phosphate groups added to it to arm it, to turn it into the active triphosphate form.
And who does that arming? Not the virus, I assume.
You do, the host cells. Your own cellular kinases have to do the work of turning the drug into the active weapon. This is a crucial concept because it means the drug's efficacy is partly dependent on the host's cellular machinery.
That feels like a potential vulnerability. Does every patient have the same ability to activate these drugs?
For the most part, yes, but it can vary by tissue type, which affects where the drug is most active. But the bigger issue that arises from this mimicry is specificity. Because these drugs mimic natural DNA blocks, they can sometimes fool our enzymes too. Not the main nuclear DNA polymerase that copies our chromosomes. That's usually smart enough to tell the difference. But the mitochondrial DNA polymerase.
Polymerase gamma.
Exactly. Mitochondria, are the ancient bacteria living inside our cells generating power. They have their own DNA, their own ribosomes, their own replication machinery. And that machinery is a little less discerning.
So the drug can accidentally get into the mitochondrial construction site?
And if it does, you get chain termination of mitochondrial DNA, you stop mitochondrial replication, the power plant shuts down.
And the downstream effect of that is?
The classic toxicity profile of this class. Lactic acidosis, because the cell can't use oxygen properly and reverts to anaerobic metabolism, fatty liver, or hepatic steatosis. And peripheral neuropathy, because neurons are so energy dependent. The older drugs like stavudine and didanosine were notorious for this. They decimated mitochondria.
But the modern ones are better.
Much, much better. Modern ones like tenofovir and abacavir are much more specific to the viral enzyme, so they're far safer. But the mechanism of toxicity is still lurking there. It's a risk you have to be aware of.
Let's drill down on 2 specific NRTIs because they show up in every regimen and they highlight some critical clinical nuances. First, abacavir. There is a huge board exam warning light flashing on this one.
A massive red flag, a klaxon horn. Abacavir is a great drug. It's very effective, but it causes a severe hypersensitivity reaction in about 5 to 8% of the population. And this isn't just a minor rash. It's a multi-system immune revolt.
What does that look like?
It's fever, malaise, nausea, respiratory distress, a distinctive rash. It can look like the flu. If you stop the drug, it gets better.
Yeah.
But if you don't recognize it and you re-challenge the patient, if you give them the drug again after this reaction, it can be fatal within hours from cardiovascular collapse.
That is incredibly high stakes, but we can predict this.
We can, 100%. It's linked to a specific genetic allele, HLA-B5701. It's a gene that codes for part of our immune system's presentation machinery.
So the rule is absolute.
Absolute. You cannot write a script for abacavir or any combination pill that contains it like Triamic until you order the genetic test and confirm the patient is HLA-B5701 negative. If they are positive, abacavir is listed as an allergen in their chart forever. You never, ever touch it. This is a prime example of pharmacogenomics using genetics to guide safety.
A perfect one. Now, what about tenofovir? I see TDF and TF mentioned constantly in the literature. It seems like the entire industry shifted from one to the other in the last decade. Why fix what wasn't broken?
Well, TDF is a little bit broken in a subtle way. Tenofovir is unique because it's a nucleotide. It already has one phosphate attached, so it bypasses that first, often slow activation step.
It has a head start.
It has a head start. But the original form, TDS, which stands for tenofovir disoproxylfumarate, had a delivery problem. To get it absorbed, it was formulated as a prodrug that broke down in the blood very early.
So you had high levels of active drug floating in the plasma.
Exactly. And that high plasma level meant the kidneys were constantly filtering it out, which caused toxicity in the proximal tubules of the kidney, essentially a drug-induced Fanconi syndrome. And for reasons that are a bit more complex, it also leads to calcium from bones, leading to decreased bone mineral density and higher risk of fractures.
So they reinvented it as TAF.
Tenofovir alafenamide is a much more stable, much smarter prodrug. It stays intact in the blood and really only breaks open and becomes active once it's inside the target lymphoid cells.
A targeted missile versus a carpet bomb.
That's a perfect analogy. You can give a much smaller dose, 25 milligrams of TF versus 300 milligrams of PDF, and you get higher concentrations inside the virus factory, the T cells, but you get 90% lower concentrations in the kidneys and the blood.
So it spares the organs while hitting the virus harder.
Precisely. It's a huge win for long-term safety, especially in an aging population with HIV who might have other comorbidities. This trade-off between potency, delivery, and safety is a recurring theme in pharmacology.
It's brilliant. Before we leave the NRTI backbone, we have to mention the M184V story with emtricitabine and lamivudine. This is one of those counterintuitive things in biology where losing is actually winning.
I love this story. It's so elegant. So emtricitabine and lamivudine are very similar cytosine analogs. They're often used interchangeably. If a patient's regimen is failing, and these drugs are on board, the virus almost always develops a specific mutation called M184V. This mutation confers high-level, complete resistance to these two drugs.
Which sounds like a total failure. The drug is now useless.
You'd think so. But the M184V mutation comes with a heavy fitness cost for the virus. The mutated reverse transcriptase is clunky, it's inefficient, it replicates much more slowly. It's like a world-class sprinter suddenly forced to run with heavy ankle weights on.
So even though the drug isn't killing the virus anymore, the mutation it forced the virus to adopt is now slowing it down?
Exactly. And here's the really weird part. That M184V mutation makes the virus hypersensitive to some other NRTIs, especially zidovudine and tenofovir. It actually makes the other drugs in the cocktail work better.
Wow.
So sometimes, even when we know a patient has resistance to lamivudine, we keep them on it, just to force the virus to keep those ankle weights on. It's a strategic manipulation of the viral genome to keep it crippled.
That is 4D chess right there. Amazing. Okay, let's move to the next class in the toolkit. The NNRTIs, the non-nukes.
Right. So if NRTIs are fake bricks that get put into the wall, NNRTIs are like sand in the gears of the cement mixer.
Okay, different approach.
Totally different. These drugs you'll see names like efavirenz, davirapine, rilpivirine. They bind to the reverse transcriptase enzyme, but not at the active site where the DNA is being built. They bind to an allosteric pocket, a little side door on the enzyme, a completely different spot.
And what does binding to that side door do?
It jams the whole machine. It induces a conformational change, a shapeshift, that freezes the enzyme's moving parts. The thumb and finger domains of the enzyme can't move, so it can't grab the next nucleotide. It's what we call non-competitive inhibition. It just paralyzes the enzyme.
It sounds effective. But Goodman and Gilman warns about a glass jaw here. What do they mean by that?
Yes, this class as a whole is fragile. Specifically, the older first-generation ones like efavirenz and nevirapine. It only takes one specific point mutation, the K103N mutation is the famous one, to completely ruin that allosteric binding pocket.
Just one typo in the whole genome.
It's just one amino acid change. Because it's not the active site, the virus doesn't lose much fitness by changing it. So that K103N mutation is often pre-existing in that viral quasi-species we talked about. If you use an NNRTI alone, or, and this is critical, if you add it as the sole new drug to a failing regimen, the virus will find that K103N mutation in days, and the entire class of drugs becomes useless.
That sounds like a very high-risk gamble. And the side effects seem very distinct, too. Efavirenz has a reputation for being, well, psychoactive.
The efavirenz dream, that's what we used to call it. It crosses the blood-brain barrier extremely well and causes a lot of CNS toxicity. Patients report incredibly vivid, often bizarre technicolor dreams. Some get dizziness, a sort of brain fog, or even severe depression and suicidality.
Sounds very disorienting.
It can be. Many patients couldn't tolerate it. And nevirapine, that's notorious for two things. Hepatotoxicity, it can really damage the liver, and severe rashes, including Stevens-Johnson syndrome.
That's where your skin essentially blisters and peels off. It's a medical emergency.
Huge emergency. With nevirapine, you actually have to lead in the dosing. You start with a half dose for 2 weeks to sort of gently trick the liver into upregulating its enzymes to handle the drug safely before you go to the full dose.
And that brings us to the liver enzymes. This class is messy with the CYP450 system, right?
Oh, it's a minefield, a complete minefield. They're both inducers and inhibitors of various CYP enzymes. They can chew up other drugs faster, or they can block the metabolism of other drugs, making their levels skyrocket. If you have a patient on polypharmacy statins, blood thinners, psych meds, and NRTIs, are a clinical headache to manage.
Which perfectly explains why the field has largely shifted away from them and towards the third major class, the integrase inhibitors, or NSTIs. These seem to be the modern champions.
They are absolutely the new standard of care. We're talking about drugs like dolutegravir, bictegravir, raltegravir, and as the name suggests, these target the integration step we talked about earlier, the physical pasting of viral DNA into the human chromosome.
So they block the paste function of the cut and paste.
Exactly. They block the strand transfer step. If the viral DNA can't be pasted in, it just floats around in the cell cytoplasm until it's eventually degraded. It can't become a permanent archive.
And why are they so much better? Is it just potency?
It's speed and durability. Clinical trials show they crash the viral load faster than any other class. But the real magic is what we call the genetic barrier, especially for the second-generation drugs like dolutegravir and bictegravir.
They are often described as forgiving. What does that mean pharmacologically?
It comes down to something called the dissociation half-life. The drug binds incredibly tightly to the active site of the integrase enzyme. It gets in there and it just stays stuck. The half-life of the drug-enzyme complex is very, very long, often longer than the interval between doses.
So it's incredibly tenacious.
Extremely.
Yeah.
Because it binds so well and for so long, the virus struggles to find a simple mutation to dislodge it. A single mutation isn't enough. It usually needs multiple complex mutations to even begin to escape, and acquiring all those at once is very rare and often cripples the virus.
So if a patient misses a dose here or there, or takes it a few hours late, there's still enough drug stuck to the target to keep the virus suppressed.
Exactly. It's essentially foolproof compared to the glass jaw of the NRTIs. That's why we start almost everyone on an integrase backbone now. The combination pill Biktarvy, which is bictegravir, emtricitabine, and TAF, is the #1 prescribed regimen in the world for this reason. It's a tiny pill, once a day, with a massive barrier to resistance.
Are there any downsides? No drug is perfect.
There's one that's emerging. We are seeing a consistent signal of weight gain. Patients on integrase inhibitors, particularly dolutegravir and bictegravir, seem to gain more weight than those on other classes. The mechanism isn't fully clear yet. It might be that their metabolism isn't constantly fighting the virus anymore, a return to health effect, or maybe it's a direct effect on adipose tissue. It's an active area of investigation.
Interesting. Okay, let's talk about the fourth major class, the scissors. The protease inhibitors, or PIs.
Right. This is where the molecular engineering gets really, really cool. So let's jump ahead in the viral life cycle. The virus has integrated, it has hijacked the cell's machinery, and it's now making new viral proteins. But it produces them as a polyprotein, one giant, fused, non-functional chain of parts, like a sheet of plastic model airplane parts that haven't been snapped apart yet.
That's the gag-pole polyprotein.
That's the one. It's totally useless in that long chain form. So the virus uses its own specialized enzyme, HIV protease, to act like a molecular pair of scissors. It goes along the chain and snip, 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 cutting it into the working pieces, the capsid, the reverse transcriptase, the integrase for the next virus.
And PIs stop the snipping.
They jam the scissors. They are designed to mimic the specific shape of the chain that the protease wants to cut, usually a phenylalanine proline sequence.
Yeah.
They sit right in the scissors blades and get them stuck.
So the new virus still buds out of the cell.
But it's a dud. It's
Right.
immature. It's full of uncut junk protein. It's not infectious. It cannot infect the next cell.
It's a very elegant mechanism. Now, you have to explain the concept of boosting. This is one of the most clever hacks in the history of pharmacology. We almost always see a PI like darunavir given with a tiny dose of ritonavir or cobicistat. Why?
This is pure systems thinking, and it's brilliant. Ritonavir is technically a protease inhibitor itself. It was one of the first ones. But we don't use it to kill a virus anymore because the therapeutic dose was way too toxic. We use it at a tiny subtherapeutic dose because it is a ferocious inhibitor of the liver enzyme CYP3A4.
The enzyme that acts as the body's main garbage disposal for drugs?
The main one. Correct. If you take darunavir alone, your liver sees it and destroys it so rapidly, you'd have to take handfuls of pills every few hours to keep the levels high enough to work. It's not practical.
Okay.
But if you take it with a little 100-milligram dose of ritonavir, the ritonavir essentially throws itself on the grenade.
It sacrifices itself for the team.
It binds to the CYP3A4 enzyme and paralyzes it. It's what we call suicide inhibition. The ritonavir is destroyed in the process, but it takes the enzyme down with it for about 24 hours.
So the liver is completely preoccupied with wrestling with the ritonavir.
And the darunavir just slips past untouched. Its levels in the blood stay high and flat for a full 24 hours, allowing for once daily dosing. We call it pharmacokinetic enhancement.
That's genius, but there has to be a price. When you paralyze the liver's main disposal system, everything else that uses that system must build up too.
And there's the catch. The drug interaction list is a mile long. Statins, antifungals, sedatives, cardiac meds, erectile dysfunction drugs, you name it. If a patient on a boosted PI takes a normal dose of simvastatin, they could get life-threatening rhabdomyolysis muscle breakdown because the statin levels skyrocket by 1,000%. You have to be a pharmacological detective to prescribe these drugs safely.
And historically, PIs changed the bodies of patients, didn't they? We need to acknowledge the history here.
We do. The metabolic syndrome. The older drugs like Antiretroviral caused profound lipodystrophy. It fundamentally altered how the body stored and managed fat. Patients would lose the subcutaneous fat in their face, what we call facial wasting, and in their limbs, making them look skeletal. But at the same time, they would gain massive amounts of visceral fat deposits in their belly and on the back of the neck.
The buffalo hump.
That's what it was called. It must have been incredibly stigmatizing. It made the disease visible. You could look at someone across the room and know they were on HIV meds. The modern PIs like darunavir and atazanavir are much, much cleaner, but they still have a tendency to raise cholesterol and cause some insulin resistance. It's a trade-off we accept for their potency, but we have to manage that long-term cardiovascular risk.
Okay, that covers the big four classes. Let's briefly touch on the bouncers at the club door, the entry inhibitors, Marviroc. This seems to be a very specific tool for a very specific job.
It is absolutely a niche drug, a very clever one, though. It doesn't target the virus at all. It blocks the CCR5 receptor on our own human T cells. This is the main door handle that most strains of HIV grab to enter the cell.
But not all HIV strains use that door.
Correct. HIV is tricky. Over time, or sometimes from the start, some strains can evolve to use a different receptor called CXCR4. So before you can even think about using Marviroc, you have to run an expensive test called a trophile assay on the patient's blood. It's a test to see which door their specific viral swarm likes to use.
And if it uses the other door?
If the patient has dual tropic virus that can use either door or CXCR4 tropic virus, Marviroc is essentially a sugar pill. It does absolutely nothing. So it's used in very specific, heavily pretreated patients.
So it's rarely first line. And what about the other one, Infubertide?
That's a fusion inhibitor. It's a peptide that binds to a viral protein called GP41. It basically stops the virus from being able to pull itself into the cell membrane after it docks. The problem is, because it's a peptide, a small protein, you can't swallow it. Your stomach would just digest it.
So it has to be injected.
Subcutaneously, twice a day.
Yeah.
And it causes very painful lumps and reactions at the injection site. It's really a salvage therapy, a last resort when the virus has become resistant to almost everything else.
So we have this incredible toolkit. But the magic is in how you put them together. We talked about the 3-drug rule, but let's look at a specific clinical scenario from Katsung to bring this home, the co-infected patient.
This is very common. HIV and hepatitis B are transmitted the same ways through blood and sexual contact, so you often see them together in the same patient. The beauty here is that some of our best NRTIs, specifically tenofovir, emtricitabine, and lamivudine, kill both birds with one stone. They are potent inhibitors of the polymerase enzyme of both HIV and Hep B.
So you treat both devastating chronic viruses for the price of one regimen.
You do. But the danger is in stopping. This is critical. If a co-infected patient stops their meds, maybe they lose their insurance or they just get pill fatigue and stop taking them, they aren't just letting HIV rebound, they are releasing the brake on hepatitis B replication.
And the Hep B flares up.
Massively. It can cause fulminant hepatic necrosis. The liver basically destroys itself in an acute inflammatory flare. It can be fatal within days. So adherence in these patients is life or death for their liver, not just their immune system. This is a massive black box warning on these drugs for that reason.
That brings us to the final and maybe most important concept of this entire deep dive, adherence as pharmacology. We said earlier that missed doses are the enemy. But can you explain why the middle ground of dosing is so dangerous? Why is taking the drug 50% of the time actually worse than 0% of the time?
It goes back to that evolutionary pressure cooker. Imagine a graph. If you have zero drug in your system, the wild-type virus grows. It's fully fit, but it's still susceptible to treatment later if you decide to start. If you have 100% drug levels, from perfect adherence, nothing grows. The virus is completely suppressed.
Okay, the two extremes.
The danger is in the middle. If you miss doses and your blood levels of the drug drop to, say, 50% of what they should be, you create the perfect selective pressure.
You're killing the weak ones, but leaving the strong ones to survive.
Exactly. You provide enough drug to kill off the susceptible wild-type virus, but not enough to kill the 1 in a million mutant that is slightly resistant. You essentially clear the field for that resistant bug to take over. You are creating a training camp for a resistant virus. You are actively breeding a superbug inside the patient.
So inconsistency is the worst possible behavior.
It is the absolute worst.
Yeah.
And if that resistance happens, if a regimen fails and the viral load becomes detectable again, you can't just add one new drug to the failing regimen. That's the cardinal sin of HIV management.
Why not? If 3 aren't working, why can't you just add a 4th to overpower it?
Because if the first 3 aren't working due to widespread resistance, adding one new drug is effectively giving functional monotherapy. That new drug is fighting the entire billion virus swarm all by itself.
And we already know how that ends.
The virus will burn through that new drug in weeks because of the high mutation rate. And then you've not only failed to resuppress the virus, you've burned that new drug class as a future option.
So you have to clean house completely.
You have to do resistance testing to see which drugs are still active, and then you switch at least 2, preferably all 3 drugs, to entirely new classes that the virus hasn't seen before. You have to reestablish the siege with a completely fresh army.
Looking forward, are we stuck with the burden of daily pills forever? Because pill fatigue is a very real thing for patients living with this for decades.
We are getting there. This is the next frontier. We now have cabotegravir and rilpivirine as injectable nanosuspensions. The patient comes in and gets a shot in the gluteal muscle every 1 or 2 months. That's it.
That changes the entire psychology of the disease. You aren't sick every morning when you take your pill.
Exactly. It decouples the treatment from the daily reminder of the disease. And on the horizon, we're looking at things like subcutaneous implants, little rods like the ones used for birth control, that could elute drug for 6 months or even a year. The future is long-acting.
But the cure, the actual excision of that proviral archive from the human genome, that remains the holy grail.
It does. Things like CRISPR and other gene editing technologies offer a theoretical hope. The idea that you could literally send in molecular scissors to find and cut the viral DNA out of every infected chromosome, but the challenge of delivering that to every single infected latent cell in the body, that is a challenge that makes landing on the moon look easy.
But considering where we were in 1985, with this being a universal death sentence, holding the line is a pretty remarkable place to be.
It's a miracle of science, a victory of systems thinking. We beat the math of nature with the math of pharmacology.
Couldn't have said it better.
It's a constant battle, but one we're currently winning.
So let's recap the journey. We've gone from the sloppy typist errors of reverse transcriptase to the brick without mortar of the NRTIs. We've seen the sand in the gears with the NNRTIs and the scissors jamming of the protease inhibitors.
And we've underscored that while the individual drugs are the weapons, combination and adherence are the strategy. Without the discipline of keeping that constant pressure, the math will always turn against you.
For those listening, if you want to see the specific resistance mutation maps, the ones that show exactly how the virus learns to fight back, open Goodman and Gilman, Chapter 64. It's like looking at the enemy's battle plans. It's fascinating.
And for the clinical tables, the dosing, the side effects, and especially those critical drug interaction tables for the PI's CatSum Chapter 49 is your go-to reference. Keep your eye on the interactions. They will bite you if you aren't looking.
Which leaves us with a final thought. We have achieved this incredible thing, durable containment. We have suppressed the virus to the point where it's a whisper. But that proviral DNA, it remains archived in the nucleus, silent and waiting. The next frontier isn't just about better suppression, it's about excision. It's about a cure. And the question to ponder is, how do we reach into the library of the human cell and tear out the pages the virus wrote?
A profound challenge for the next generation.
Keep learning, keep reading, and stay curious. Thanks for diving deep with us.
Thank you. It was a pleasure.